Good evening, Maryville College, and welcome to Micro News. I'm Gabriella Lister, and I'm here to give you important updates in microbiology. The mysterious pathogen called Elisabethingia anopheles that left 18 people dead and more injured in Wisconsin 2 years ago has struck again, this time in Greece. Though only known to exist in the midgut of the Anopheles mosquito, recent investigations have indicated that waters in hospitals may act as a reservoir. To find out more, we have here with us Maria Chirizzi, head researcher on this breaking news. Welcome, Maria. What exactly can you tell us about this new pathogen that's been detected in Greece? Well, Gabriella, the cause of this pathogen is a bacterium called Elisabethingia anopheles. It is capable of wreaking havoc due to its ability to exacerbate current diseases and enter the bloodstream, and it is especially dangerous in those with a weakened or suppressed immune system. My team and I tested 11 hospitals throughout central and northern Greece and found 3 instances of bacterium belonging to Elisabethingia genus, 2 of which we believe belong to Elisabethingia anopheles. Fascinating. Could you please walk us through how you and your team went about collecting these water samples and what tests you used to isolate the bacteria from the Elisabethingia family? Certainly. We obtained 243 samples directly from the 11 hospitals that we investigated. Each of the samples came directly from the tap without any disinfection. We then filtrated the water samples onto a paper disc called a membrane to collect the bacteria. To isolate the bacterium from the Elisabethingia genus, we performed a series of biochemical tests to isolate the bacteria that were gram-negative, non-fermenting, non-motile, and aerobic, meaning that they cause infections in humans, they're unable to break down lactose, unable to move on their own, and require oxygen for respiration. These tests included the use of differential media and tests for specific chemicals that are necessary for aerobic respiration, such as cytochrome C and catalase. Once these bacteria were isolated, we were able to sort them by their chemical makeup using a matrix-assisted laser desorption ionization time-of-flight mass spectrometry, which we refer to as the Maldi-Toff machine. Once we obtained the data from the Maldi-Toff machine, we were able to match our data to the Maldi biotyper library to determine the genus and species of the bacterium. From this, we were able to identify 3 bacteria belonging to the Elisabethingia genus, with 1 bacterium being identified as Elisabethingia miracola and the other 2 being identified as Elisabethingia meningoseptica. Due to the newness of this pathogen, my team and I were concerned that our tests might misidentify the bacterium and classify it as a different species within the Elisabethingia genus. To investigate this, we isolated the DNA of the bacterium using a QIAMP DNA mini-kit, and we amplified the 16S rRNA region using a polymerase chain reaction. We then sequenced the 16S rRNA genome and used the sequence on the Maldi-Toff data to identify the bacteria by inserting all of the information into a blast search of the MicrobeNet CDC reference laboratory. In doing this, we were able to update the Bruker library and correctly identify the Elisabethingia anopheles bacteria that had been incorrectly identified as Elisabethingia meningoseptica. So, what does all this mean for the hospitals and their patients? Our study has provided further support that Elisabethingia anopheles is present in hospitals and that these waters may act as a reservoir. Furthermore, our experiment shows that more research is needed on both Elisabethingia anopheles and the Elisabethingia genus to properly understand where the pathogen comes from, how it is transmitted, and what we can do to prevent infection. Due to the inaccuracy of the testing methods, it is plausible that patients that become infected with Elisabethingia anopheles would be diagnosed with the wrong strain and receive an incorrect treatment. Though there were no reported deaths resulting from an Elisabethingia anopheles infection, we do have reason to believe that infection with Elisabethingia anopheles assisted in disease severity for several patients. That's all we have for today. Please join us tomorrow as we discuss the dangers of eating salads.